I start again. So today I want to discuss with you about my research, give you a little bit of the flavor of the question we are asking, and also how we interplay the experimental and modeling approach. So I'm going to talk about hematopoietic differentiation at the single cell level. So to start with, what is hematopoiesis? So hematopoiesis is the process that leads to the production of immune and blood cells. So on the right you get all the names of the different cells that are produced from the textbook. It's supposed to come from a hematopoietic stem cell, which are called cells, HSCs. And then those HSCs are supposed to self-renew and give rise by differentiation to this multipotent progenitor. So they are called multipotent progenitor because they can give rise to all the cell types. And then there is a major split between the lymphoid cells, so all those are lymphoid cells, and those that are called myeloid cells. And there are some cells in the middle that are called dendritic cells that are produced by both branches. So this is also called a tissue, but it's a non-solid tissue, it's a diffuse tissue. Those cells are present in multiple organs. And so maybe to start with, I want to ask you, which type of question do you have in mind when you see this picture about the tree? So I'm going to give you 2 minutes to think and write about this. So I show you the tree again. So when you see that tree, what are the questions you have? Okay, I'm sure. So you are many in the room, so I'm not going to ask each of you. So I made a list, and maybe if you have a question that are not on this list, which is most probable, don't hesitate to raise your hand and ask your question. So traditionally, people have been asking questions about which genes are involved in this process, so which are the genes that govern the differentiation. Also, they have been asking questions about organ location because the hematopoietic system is a diffuse tissue, so the cells can migrate more or less everywhere, not completely everywhere. So where does every stage is happening? There is also a lot of research about the influence of the microenvironment on this differentiation. Bit more quantitative questions that we actually don't know the answer. How many divisions? What is the rate of production of those cells? Because some of those cells that are produced from the hematopoietic cells can respond to infection, so they are the immune cells. So upon infection, they can proliferate, they can divide. So or is the production changed by those perturbations, we can say, and also importantly, after a perturbation like infection, how does it return to equilibrium? How is the decision made, and where is the decision made? Is also a question that you can ask. And also, is it the same process for every cell or the same? And I could continue the list, so I don't know if someone wants to add a surprising question to the list. You got all those questions? Okay, well, doesn't matter. What I'm interested in is whether the process is the same for every cell, and or one cell is taking this decision to become a certain cell type and not other cell type. So maybe to continue in the introduction is why do we care about studying this process at the single cell level? And I think it's because when you study differentiation, if you would do a population assay, you cannot distinguish between these two scenarios where one cell type and another cell type are produced either by the same precursors so meaning the same cell is giving rise to the two cell types, or whether it's actually an heterogeneous population and one cell is giving rise to one cell type and another cell to another cell type. And so the only way to distinguish between these two scenarios is to actually follow the cells at the single cell level. So if you would put a tag, like here a flag in the precursors, then you can distinguish whether they come from the same progenitor, meaning the same cell, or if they have a different origin. So by putting a tag in the cell, which is called lineage tracing, you can get information about the kinship between two cell types, but also about the heterogeneity of your initial cell population. So in my lab, this is what we do. So we do this type of tagging with a method that is called a cellular barcoding. So how does it work? You get a plasmid vector with a random sequence of 100 nucleotides and a GFP reporter. And we get those sequences in the library, and for the moment we have 2,500 different barcodes. So all the experiment I'm going to show is with this diversity, but we also now have libraries that are 100, 1,000 different sequences. So each of these vectors are encoded in an antivirus. And then, so how do we use it? So, for example, you're interested in hematopoietic stem cells, so you take a population of interest. You have your barcode library, so every virus has a different tag or barcode. You use the antivirus to infect the cells, and then the antivirus is integrating your tag into the DNA of the cells, and you get a different tag for every cell. Then what we do is that we split at least in two, and we inject into mice that are conditioned to receive the cells, so meaning we do a treatment to remove some of the cells, the hematopoietic cells from the mouse to make room for the cells we're going to inject. So we inject, and then we wait some time, and the cells divide and differentiate. And then we take, we collect different organs from that mouse. We sort by fax different cell types. And in this cell type, we do PCR and sequencing, and then we retrieve the identity of the tag in the different cell type. And then from there, we can start to compare whether the tag are shared between the cell types, whether the diversity of the tag is the same within one cell type and another. So to test whether you have followed the protocol, I have a quiz for you. So here is an experiment, so it's the summary of what I showed before. You have the virus that infect, and we collect these two cell types, A and B, and we have three barcodes, the blue, the yellow, and the purple, and these are the cells that are made in each of the cell types. So I'm going to ask you to raise your hand for the answer to the question. So how many cells of type B come from barcode 1, which is blue? So is it 0? Or is it 2? And so those who haven't raised your hand, what's going on? You can do it again. So is it 2? Or is it 0? Okay. So it was 0. Then again, how many cells come from barcode 2, so the yellow? So in total, is it 2 or 4? Okay, so in total from cell A and B, you have 4 yellow cells, so it's 4. Well, we can continue like that. And I think most of you got the principle of it. So that's exactly what we are doing when we receive the data from sequencing. We are compiling this type of information. So maybe one question you have about our system is, how can we be sure that there is one barcode per cell? Because we use the lentivirus. So if we will get 2 barcodes per cell, it will look like that. And in terms of data, it will look like we had 2 different progenitors. So this is actually messing up with our quantification, not changing the information about the cell fate, but it's just counting double something that was one event at the beginning. So we cannot avoid it, but what we do is that we reduce transduction efficiency, so meaning we decrease the ability of the virus to infect. Like that, not all the cells are infected, but the chance to have one virus in one cell is increased. Okay, something else that can go wrong is that if we get 2 times the same tag in 2 different cells, then it will look like we had one cell that is producing everything. So this will give us the information, the impression that we get more multi-outcome cells than what we really have. So for this, what we use the library that is a bigger size than the sample. And also, we always have a control. So as we always inject at least in 2 different mice the same batch, we can compare the barcode that are shared between 2 mice. So that's here you see a plot where you have the 2 mice, and every dot is a barcode, and you see there is a few barcodes that are shared, but the rest are on the axis. So this means that there was at least 2 cells that went one into each mouse with that barcodes. So we know this is happening, but in general it's around 5% in all the experiments. So we always check for it, and then we keep it in mind for our conclusion. Yes?
So do you use multiple copies of the same barcode during the infection?
Yeah, so the library, the library when it's produced, it's not we take one virus of each and we make a pool. We had to expand everything together, so there is a different frequency of that virus in the pool, and actually we have sequenced it, so we know we can compute the probability of that barcode to infect. And yeah, you can predict actually this result quite well. We haven't gone more far than that because, I mean, what is important in the end is the barcode that gets in. And you see in most of the conclusion, actually, this is going against what we're going to conclude. All right, so with this system, what we have been, one of the first questions we have been asking is whether a progenitor that is called, that is a multipotent progenitor. So it's this progenitor. It's a progenitor that can produce everything except red blood cells and platelets. We don't really care about the name of that progenitor, but the question was that if you put a barcode in every of those cells, all those cells actually the same, or they're really producing everything, because that's what you expect from a multipotent cell. So for that, we sorted those progenitor by fax. We label them with the barcode, inject them into a mouse, and collect different cell types from the hematopoietic system in the spleen and the bone marrow. And so this is the result we have. So on this heat map, every row is a barcode. So one progenitor with one barcode. And every column is a cell type. So you don't really see it well, but here are the dendritic cell type. Here you have the neutrophils, the monocytes, and here the B cells. So, well, what do you see first? And sorry, I forgot to say, red means we find a lot of reads, so a lot of cells with that barcode, and black means we don't detect that barcode in that cell type. So what do we see? We see a lot of black, actually, meaning that progenitor, we didn't detect any progeny in those cell type. And so this means actually that all these progenitors, they are not actually giving rise to every cell type, as you could think from their definition as a multi-outcome. So not every progenitor makes every cell type. And be careful here, I'm not talking about potential, because potential is what they can do, but what we measure is what they are doing, so what they are actually doing. So we measure the outcome. We cannot say anything about their potential because we cannot take them back and put them into another mouse. So we can only do the experiments once, so we can only say something about what they're actually doing. So here we can say that those multipotent progenitor, I mean that's the way they were defined, are not multi-outcome. But still they are very heterogeneous. You see they make different combinations of all those cell types. But we found that this pattern of production was reproducible between mice. So the proportion of barcode that produce either the three cell types, myeloid cells, oncolytic cells, and B cells, these are the three group. The one that produces the three is only 10%, and the standard deviation here is between mice. And then you see that you get different combinations. They can produce two of them or only one, and with different proportion. And what was striking also is that a lot of those progenitors only produce one cell type, the oncolytic cells. Okay, so from this first experiment, we conclude that the progenitors are not the same, but there seems to be some group of pattern of production between those progenitors, and that is represented here. Well, we did the same with the hematopoietic stem cell, the one that's at the top of the hierarchy. And that's what we obtained. So you see it's quite different from what we have seen before. And again, I'm going to ask you what you think the conclusion are this time. So are all the stem cell equivalent? Yes. Raise your hand if you think yes. And no. All right. And the next question is, are the stem cell more multi-outcome than the previous progenitor type? Yes? Or no? All right. Well, I think from so it's quite obvious we get different patterns, but it's very different from what we see before, because now we get those lines that have a color in all the different cell types. So we do get some progenitor that produce everything. I think if you compare in terms of proportion, it's more than what we have seen with the multipotent progenitor. And also the cells produce much more cells than the other one. And we also get some that only produce one or the other cell types. But in general, stem cell, I mean, this reproduces well what was known about stem cells. So people were doing single cell transplantation, so they were injecting one stem cell into one mouse, and so it's a lot of work. And they also found that one stem cell is enough to reconstitute the whole hematopoietic system, but some actually produce only one cell type. So that's what you see here. Oh yes. And so from this, what do you think? Where is the heterogeneity appearing in the stem cell before, in the differentiation before the stem cell or after the stem cell? Who says before? Who says after? And both. Yes, so I think what we have is more that you get a little bit of heterogeneity already at the single cell level in the stem cell, and this heterogeneity is increasing when you reach this multipotent stage. Okay, what can we not say with cellular barcoding? Because I get this question a lot. So can you say something about the point of commitment? And the thing is that we can't really say, like from one experiment, from one progenitor, because we put the barcode in at the beginning, and then we only see what this barcode is producing. We have no information about the intermediates. So we cannot distinguish these two scenarios where here the commitment is only occurring after this stage, and here it's already at the first division. It was decided to be red and green. This we cannot answer with one experiment. We may be able to answer it by sorting these different stages and injecting them, but of course when you do that you don't know how much you're perturbing the process. And so can you say something about the differentiation, the fact that the cell is changing phase? So there are different scenarios. You can think of a simple branching situation, or the differentiation where a cell is becoming green and then back to progenitor stage and then becoming red, or transdifferentiation, meaning that you change directly from differentiated cell type, not with the progenitor. So this we cannot say anything with barcoding again, because we only get the results. So those three scenarios are kind of equivalent for us. We find barcodes, the same barcodes in the green and the red in the three processes. So going back to the tree, so from this multipotent progenitor, we have this pattern that they produce different quantity and different proportion and different combination of cell type. So the question is, can we say something about this tree from this proportion? So one very naive approach would say yes, because you get so many dendritic cells, so those cells, you just need to add an arrow and that will produce much more dendritic cells. But I decided to take a more quantitative approach. So for that, first what I did is to binarize the data, so it's the same data than here, but then if a barcode is I will just look at presence or absence of that barcode, so then the different proportion look like this. And so why do I do that? Is because it's in a way it's more robust data, because if you really take into account the number of cells, you need to be sure that you sample well all the cells that are present. So for example, it's like if I want to know in this building, the one that is on the left, how many persons are speaking French with a Dutch accent, for example, I would have to go into, I mean, if I really want to answer this question correctly, I will have to go in every room and talk in English with everybody and try to recognize what is a Dutch accent and count them. If I want to answer the question of is there at least one person speaking English with a Dutch accent, I just need to go into my room and meet my postdoc Tamar. So that's a much more easy question actually. So we decided to do the same, because here saying do I have at least one cell with that barcode is it's more robust to sampling than if I would have to say how many cells exactly. So we build a model based on a stochastic model based on branching processes. So I'm not going to go into detail of the math. I mean, some of this is published, so you can also go and look for it. But the idea was to model it as a network. So you start with a cell that can produce the three fates we have seen. So becoming an M, a D, and a B. And these cells can divide with a certain probability or can differentiate with other probabilities. And so when the cell is differentiating, the cell is losing one potential. So becoming, for example, an MDB to a DB is losing the potential of producing cells that are M. And so the next step, you can do the same, divide or differentiate. So if you start, let's say, with one cell with a red barcode, that cell can differentiate, like losing the M potential, and then, for example, divide here with a certain probability, and then differentiate and produce a D and a B. If I do that again with a cell with a blue barcode, that cell, for example, become D, and that's it. So then I can count how many colors I have in every cell type, and I can produce here. I can produce. I can compute. Actually, we were able to solve with a finite expression. We could solve for a given pattern what are actually the set of probabilities that will give rise to this different proportion of barcodes in different cell types. So for that, we had to make an assumption, and I think it's important to discuss them. We had to assume that the events are independent. That's the case for most of these stochastic models. So what does that mean in terms of biology? It means that if I produce a B cell, I mean I cannot give any feedback to the progenitor upstream to say I have produced some B cells. So there is no feedback possible, and you might think it's something that is happening, like in the hematopoietic system, it's not that you're going to produce only one cell type and there is no regulation. But because we use this irradiation, so we remove some progenitor from the mouse, and we are in a stage where there is a lot of space, those cells are pushed to produce. So I think with those experiments, at least, because we make space for the cell to divide, there is not a lot of feedback yet. It may happen later when you go back to equilibrium. Another assumption is that there is no death. Remember that for later, it will be important. And also, I think the reason why we decided for that is that there was no way to measure depth. So it's a simple, because we cannot measure it, we decided not to include it into the model. Okay, so to make this a little bit more concrete, I propose that we do this game is to do with M&Ms and dyes what the model is doing, like that you can get a feeling of what is going on. And this we will do it with the classical model. So this model is the one I showed you at the beginning. So that's what is, let's say, in the textbook. So now it looks like that in this model. You get a cell that can produce the 3 cell type. Then you get the myeloid and the lymphoid split. So progenitor that can only produce myeloid cells, and progenitor that can only produce lymphoid cells, here B cells, and then the dendritic cells, as you remember, can come from both. So the pattern we have to mimic is this. So what you will do, you will get the network, and then the dice to mimic the different probability, and then with the dragibus, you get different cell with the same color. So what you do at the beginning is you start with one dragibus of one color, you roll the dice. If you get something that is less than 3, then it means that it's going to divide. So you take another dragibus of the same color, that's your 2 cells. And then you draw the dice for each of them, and if they are if you have 3 and 4, then it will go to the left, so it will become a myeloid, or it will go to that stage, myeloid progenitor, and if you get the other possibility, you go to that stage. And you do that for every cell. And I said no depth, so you're not supposed to eat them now. For the moment. Okay, and so the idea is that you do that with a certain number of progenitor, and then you will see where do they end up with the colors, and you can try to see whether you will arrive to, will be able to reproduce that pattern or not. Yeah, and you can pair like by 2 or 3. You can, so you get okay, you already get one. Please ask me any question if you need. Yes. Yes. I'll do it. So I start with a progenitor that is red. So this one starts there. Then I roll the dice. When it's 3, so then divide. Get 2. We roll the dice again. It's 6, so it's going there. So it's 5 like this. Then I continue. So it's going to divide. When you continue like that. You see? And in the end, you will maybe arrive like this. And this is count as one progenitor that gives only B. So one on the left. Okay.
Did you get that? No, nothing higher than good so far. But then we all eat. Yeah. But you can invent new ones. Are they only physical? Oh no, they're not.
Do it again and divides again.
Divide again.
That means it makes 4.
I think only
Yes,
this
I
one.
will
So we
find
go on with
another
one cell.
blue one.
This one is differentiated again.
Okay.
So,
This one
Go to the left.
divides again, right?
All of them? Or just one?
I think it's just one. Just one. Okay, so
Okay, now we have to do it again with 46 cells.
go to the right.
We don't like making
One,
this.
so divide with
5.
divide the last one.
What is this divide?
When so it divides, wait.
Yes.
Okay.
4, so that's another division.
Now, okay, now yes,
It goes left, right? Yeah.
seeing this one.
Who goes left?
So
Okay, let's try this. Let's see what we're getting from.
We have the thing because we have our
Choose one, then you do a roll and see what it does. If it's left, right, or add one more. And then you choose another one.
Ah, no way.
And you just go to the final.
I think so, yeah.
Well, we divided loads at the beginning. And so we had lots at the beginning. And then
okay, just follow this one again. Oh, I know.
Divides again. Let me see
you
what
got 3 to go left.
Yes.
This go left? I guess it doesn't matter.
Okay, so let's. So now
Can we also mutate them because I don't have any pink ones?
this one and this one.
Ah, yes.
Divides. Well, we'll divide in the same color.
We have to get
Okay, we'll divide in the same color.
I don't think they can.
Yes.
so divide again.
So 5. So go to the right
Oh, no
and
way.
then just follow this one, isn't it? So this is finished.
And again.
So
They can't divide
let's
again because
see
we don't
this
have
one.
any more.
So
Ah, no way. Okay, so we have to
okay.
pick one in the first place.
Left.
Oh, we do. Yes, we do.
Yes.
Really a lot of them.
Okay.
How do we get new ones here?
Okay,
Oh, we
here.
don't.
We have a B cell.
It's just a pre-divided thing.
Yes. Yes. So now we're with this. So this is divided.
Cool.
Yes.
That's another B cell.
So 5. It's go to the right. Okay. Okay. I'm getting. So I just this one. 2 divides.
And another one.
Okay.
We're building an adaptive immune system.
And we're just following this one. So no, okay, divide. Okay, so I guess.
Okay, which one shall we add? The blue one? I think the purple. Oh yeah, but purple we already
So
have.
4 is
Let's have a blue one. And then we'll have the yellow ones to go with this one, and then we can do purple later.
we divide a lot. So I think that as more we divide, we just get more like.
Yeah. She still has some there at the beginning. The first one.
Oh,
Yeah.
we need more bags.
Yes.
Okay, we'll have purple ones.
Maybe
instead of
that's
yellow
like it
ones.
divides lots in the beginning.
I don't know.
Let's see what happens.
Alright.
They're all equally like the 1, 2,
This one, and we just finished. Okay, so
Which is another division.
okay,
These
just
cells
pink
keep dividing like crazy.
one. Divide again.
Okay, another one.
6, so right. So the black one.
Division.
Left
Advise.
and right.
Okay. Okay, we finished.
Ah, okay. Okay, let's go for black now.
We dividing this hand.
We are wondering.
We don't have enough candy to have them divide very much. So
Why share what?
Type N plus D plus 1. because
Really have it here.
Yellows.
Ah,
We just use reds, huh? Instead of orange.
as long as you remember,
Yeah.
Because
then you can all
we're running out of colors.
We're gonna bias all your statistics. They're all the same
attributes
type.
were not made well actually. You should order some.
M plus D plus V at the final. Because if we just follow one until the end, it's until or we are doing something wrong.
They will eventually go to the end.
Yes, but if you have something that is M plus B plus D, it has to divide, otherwise it cannot produce the tree, right?
Yes.
So if it's not dividing, the cell is not dividing, then it will only produce one. So
So as we only will get the M or D or B.
yes,
We can
exactly.
exactly divide.
And then this you register it as something giving only one of the cell type.
Only M or D or B.
Yes, exactly. And then once you get the division, then you can be able to produce at least two. Maybe it will produce the same, we don't know, but it has the potential to produce two at least, maybe at some point. If you get a division. Not clear.
Yes. But it's
How many should we start with? We only started with one.
Oh, you can add more. Yes. Different colors and.
What's the ratio at the end? It's like a quarter, a quarter, or
Yeah, it depends a bit on your simulation, right? It depends with how many you start and so on. On average, yes, it should be. Yeah. Yes. So, what do you think about that then? Okay.
We did a few of
Yeah, more. You can start again, right? What you can do is to record the fate you had and you take them back and you start again, right?
when you just abstract the first one doesn't have enough time to just divide, so you just record it. But then finally
And then if you want, you can also start playing by changing the probabilities, right?
not
Making it more likely to divide or to go one way or the other. Yes, exactly.
Changing. It might stay like this for a long time, but there's nothing to stop it changing eventually.
So can I say something? So if you don't have any more colors, you can just write on a piece of paper how many you get in the different fate and do it again. If you think you want to change some of the rules, some of the probability, you can also play with that to see if, what are the rules that are giving rise to this pattern that we have here. So remember, a lot of these, not so much that give everything, in different proportions. Let's say 5 more minutes, maybe a bit less. C'est une bonne question. C'est une bonne question à poser devant tout le monde, mais sinon, parce qu'à présent c'était le modèle standard. En fait. Et il y a quand même ces papiers qui sont sortis où ils ont fait du lineage tracing aussi in situ, donc sans transplantation, et ils trouvent des dynamiques très différentes. Donc a priori, on affirmerait qu'il y a une grande différence. Mais bon, ça fait que 1 an qu'on fait à peu près ça. Donc, et ils utilisent un système génétique avec un transposon qui saute à différents endroits du génome, et l'endroit où il s'intègre dans le génome devient leur barcode. Et c'est inductible. Non, ils l'ont partout. Ah non, ils l'ont spécifique dans la moelle pour les cellules. Hematopoietic. Non, ça saute quand c'est induit. Donc c'est pas que quand c'est toutes les cellules, mais avec le temps, on voit les cellules les plus différenciées, elles meurent. Et après, au fur et à mesure du temps, il reste de plus en plus de cellules.
So 1D. Okay. 3. Let's start again.
So it's time. So I'm going to give you the answer, so you can eat the dragibus now. It's okay. I saw some of you already did, but it's okay. So basically, with the computer, we do the same. So because we have for the biologists, what you did now is like you do a simulation just in real life. And so you could solve this problem by simulation, but because we were able to solve with equation what should be the pattern, then we just take different combination of parameters and we just look for the best combination of parameters to give those combinations. So that's the best fit we get. And so actually, what is happening, it's something strange, is that it's able more or less to reproduce all the different proportions with that model and one set of parameters. But this, actually, the M plus B, you never manage to make it close to what we found in the data. So first, you may think, oh, it's a small proportion, it doesn't really matter. But actually, it matters a lot because this is a result of the structure of that model. So if you look at the textbook model, it's very unlikely that you make a M and a B without making a D, because you have this thing here. So with the probabilities, if you want to make, so it's not only the M plus B that is not really well fitted, it's the combination of M plus B, M plus D, and D plus B. So if you want to make that many D and that many M and B and MD, then you never manage to make the right proportion of M plus B. So we could reject actually that the data can be made from this model. So with our interpretation of the classical model, we reject that model. So then we looked for another model. So we went back to the full network, and first thing we did, so there we have 12 parameters. So what we did is whether we can fit the data with that model. So yes, we can, but then we have several combinations of parameters because we have too much degree of freedom. So there is different type of combination that can explain the model, but at least it said that there is enough links to explain the data, because it could be that it's not, it will not work already. And then what we did is that we started to delete one link by one link. And then every time you do that, you get actually a rejection of the model. So this is telling you that you need all the links to actually explain the data. Well, so then we thought of maybe we do some assumption to reduce the number of parameters that you have in the model. So we made this assumption of equal loss of potential. So what's going on is that if you are MDB and you become a DB, you lose the potential to be M. And so we fixed that this probability is the same of losing M from a MD to a D. So of course, because the probability they sum, it's not exactly the same value, so it's in proportion. So the losing, so here it's losing B in these two situations is the same rate, this AB, and it's over the sum of the other arrows in the network. So with this, now we have 7 free parameters. So we reduce the dimensionality of our model. We have 4 division rate and 3 differentiation rates, losing M, losing B, losing D. And so with that, we were able actually to fit the model quite well. It's the second bars. And it was not rejected with a statistical test. Yes?
So surely your conclusion will depend on the number of samples, the number of cells that you are using. So
yeah, so we had 300 cells, so barcode per mouse, and then we had 4 mouse, and so each time we were fitting with on the sum of all the different mice, and then testing per mouse if we still reproduce the pattern per mouse, and it was the case. So it's 4 times 300.
And have you tried, for instance, to do the same analysis with half the samples?
Yes.
To
Yes.
see how robust
Yeah.
it was?
Exactly, and it was still robust. Actually, you need to delete more than 60% of the data to lose this conclusion. So basically, at this moment, you have almost no data. I mean, the pattern is very different. Yes, we checked that for the robustness.
And do you think that by having, let's say, 10 times more data, which may be not possible, but do you think you may...
Well, you will just make
learn
this...
something more
Yeah.
or not?
Well, we did simulate that, and it's just that the error bars are becoming smaller, and you're more. I think it will. What you will gain by having more data is that you're more sure of the parameters themselves, that you have estimated the right value. Because at the moment, the conclusion that this model is fitting well is robust, but then the value of the parameters, you have a lot of uncertainty about them. So I will not. Make any biological interpretation, for example, of the division rate or the probability of losing one potential. Maybe if you get more data, this will become more accurate. All right, so in terms of biology, what was interesting in this model is that we have all the link. So it means like all the possible paths seems to be necessary to explain our data, and this is then the picture is very different from the classical model you get for hematopoiesis. But what is also funny is that if you look at those values, then of the parameters we get, then the main decision that is taken from this MDB stage is to become an MD and a DB, like in the classical model. So the most probable path is the one that you have found in the classical model, and then you get this very infrequent or improbable path, but still they are necessary to explain the data. And I think this is one interpretation of that I have is that because those experiments were done at the population level before, they were capturing the most probable path, and it's only when you start to look at the single cell level that you start to see this very infrequent path. Well, then, but the dendritic cell, they were supposed to come from these two, and now they can come from at least 5 different paths. They can come different here, from this, and well, there are 2 combinations of that. So it's a stochastic model, but I'm not saying anything about the cause of this heterogeneity. I don't know if it's stochastic or if it was imprinted. I'm just describing the process with that. Okay, so to conclude, we have seen that multipotent progenitors are not multi-outcome in vivo, but that there is an heterogeneous class of outcome. And I can tell you that I've worked with other progenitors and I've found a similar pattern. The classical model cannot describe our barcoding data, and more paths than in the classical model are required. An equal loss of potential can explain our data. And so I have done some experiments. I don't have time to show you today, but we have shown that actually this heterogeneity can come from a combination of niche effect and stochasticity or some decision before clonal expansion. And so to go back to the tree, just a minute before we go, so we've seen, if I would take the same picture, now we think we have different MPPs. It's not that we get one that can make everything. We have also shown actually that this doesn't exist. So this, so here you get the M or those cells, the myeloid cells, and E is for erythroblasts, so the red blood cells. So we're looking at these cells and these cells. So the CMP, which stands for common myeloid progenitor, is supposed to produce both. And what we see here, every dot is a barcode, is that actually most cells produce one or the other, and very few produce both. So, so you have the different proportion here. And so from that, we said that actually here you have 2 already different progenitors that are already made that the decision, and then the decision, maybe the common progenitor is actually already in this pool earlier. But so, I think we could continue by doing that, and I think that's what the lab is doing. So now we are trying, because we know that part of this tree is wrong, we are trying to explore different aspects of it to try to decipher the tree. And what I would like to leave you with is maybe a more philosophical reflection about this tree, is how shall we interpret actually the differentiation tree? If you take a classical picture, is it like every cell can do everything in the tree? And I think we have shown quite clearly that it's not the case. But is the tree, is it a good idea of thinking about the tree? And I think one proposal that I have at the moment is to think of this representation as a summary of all the possible trajectory, more like the union of different possible trajectories that will give rise to that. So when you look at the tree, it doesn't mean that every cell is doing what is in the tree, but it's more the union of all the possible trajectories. And of course, I mean, as a last remark, this tree has no really quantitative rates. So you don't know how many divisions, how many cells are produced. So I think the field really should more work to a more quantitative description of the tree. Okay, so with this, I just want to thank all the people. So most of the work were done while I was a postdoc, both in the Netherlands Cancer Institute in Amsterdam, in the lab of Ton Schumacher, and in the lab of Rob de Boer in Utrecht University. All the mathematical model and the derivation were done by Ken Duffy in Ireland. And I also want to thank the people in my group and you for your attention, and I'm happy to take any question.